Hi, everybody. Until now, you've been learning about pre-processing fMRI data and doing first-level analysis at the subject level. But eventually, if we want to say anything useful about how the brain works in general, we need to do group-level or second-level analysis. And that's what this lecture will be about. I'll give you a brief overview of the logic behind second-level analysis and about the differences between fixed effects and random effects in this context. I will then show you how all of this is implemented in SPM, and I will end with some recommendations for analyzing fMRI data. Now let's start by looking at the activity of a single voxel in the brain for a single participant. On the x-axis of this plot is the time measured in scans, and on the y-axis is the change in bolt signal over time. So this just to check your general understanding, think about how you can actually calculate the actual time in seconds of this activity based on this reflection of the activity in scans. All right, now each of the red dots represent a measured scan for this voxel, and the green line represents the fit of the general linear model, which you will have learned about in a previous lecture. Now, as you can see, the model fit is pretty good, as in the green line follows the red dots very well. And these data were recorded during a very simple visual experiment. And in this experiment, there was a rest period during which no stimulus was shown, followed by the presentation of a visual stimulus. And followed by another rest period, another stimulus, and so on and so forth. So these are likely data from somewhere in the visual cortex where you can clearly see this periodic response to every visual stimulation. And this is also why the GLM fits so well, because the brain response in this specific context is pretty simple. And with more complicated experiments and designs, the data will likely look more complicated as well, and it might be harder for the GLM to fit the data. Now we can calculate the effect size for this specific voxel and for this participant by looking at the difference between the rest period and the stimulation period. And in this case, that would be 4. So for this participant, for this specific effect, the effect size is 4. Now, if we've normalized our subject data to all be in the same standard space, we can look at the same voxel in another subject and calculate the effect size again by fitting a GLM on this subject's data. And in this case, the response is lower, so the effect size for this voxel for this subject is also lower. In this case, approximately 2. And here's another subject, which again has a slightly bigger effect size of 4. Now, this study had 12 subjects, which nowadays is way too small for a group study, but given that the design was so simple, we might still be able to find reliable effects at the group level. For our voxel of interest, in order to test whether there's a significant effect, we write down the effect size for every single subject over here. Now, to determine whether there is an effect of our experimental manipulation, in this case, a visual stimulus versus rest, we need to determine whether the average of the group is significantly above 0. Now, in order to determine that, we can just do a one-sample t-test on the effect sizes over subjects. To do that, we first have to calculate the mean, which in this case is 2.67, which means that on average, visual stimulation had an effect size of 2.67 in this group of participants. We then need to calculate how variable this effect is between subjects, which we determine by calculating the standard deviation, which in this case is 1.07. Now, if you remember your stats from your undergraduate, this would indicate that 64% of participants have an effect size between 2.67 minus and plus 1.07. Now, for the t-test, we need to furthermore account for the number of subjects that we have, which indicates how trustworthy our estimates are, right? If we have more subjects, we can believe these means and standard deviations more. And we do this in this case by transforming the standard deviation to the standard error of the mean by dividing it by the square root of n, which in this case is 12. So 1.07 divided by the square root of 12 gives us a standard error of the mean of 0.31. Now the t-value is then calculated as the mean divided by the standard error of the mean, which in this case gives us a t-value of 8.61. And that's quite high. Now, to determine the p-value, we can just look up the t-value in the student's t-distribution. And in this case, a t-value of 8.61 gives us a p-value of 10 to the minus 6, which is very small and definitely below the cutoff of 0.05, which we usually use in for stats to determine significance. significance. So this means that we can be pretty certain that visual stimulation had a significant effect on the activity in this voxel. Now, it is important to mention that if we do this test over the whole brain, so in many different voxels, we would also have to correct for something called multiple comparisons. And this is because if you test often enough, you might just find a significant effect purely by chance in some voxels. And correction for multiple comparisons ensures that we take that into account when we interpret our findings. For example, by lowering the alpha so that a p-value must be lower before we consider it significant. There's different kinds of corrections, and we'll get back to that later as well. Now, this type of analysis is called a random effects analysis because we compare the group mean to the between subject variability. If the group is more variable, the effect is less reliable. And the t-value will be lower and the p-value will be higher. Now, this approach is also sometimes called the summary statistic approach because we summarize the response of each subject by a single statistic, their effect size. And there's other ways of doing it where you also take into account within subject variation, for example, Bayesian approaches, but this is the summary statistic. We just look at the mean or the effect size of a single participant. Now, another type of analysis that you may have heard about is a fixed effects analysis. And this type of analysis ignores the fact that each subject is a random sample from the population and therefore prevents us from drawing conclusions about the population. And that is why this is not recommended for most neuroimaging analysis. But I will still explain how it's implemented in SPM so you can appreciate the difference between the two. Now, instead of focusing on the between subject variability, which is what we do in a random effects analysis, we now pretend that each scan is one subject, meaning that we have 600 subjects in total, where we just concatenate the scans of all the subjects. We can now calculate the variability of the data per subject and average this to get an average measure of variance. Now, variability is operationalized as how well the GLM can fit the data, and many factors influence this. Scanner noise, number of scans, etc. And we can calculate this as the difference between the samples and the model fit, and then square it, sum it, and then take the root, which gives us the root mean square error for that subject. We do this for every subject, and the worse the fit, the larger this measure. And next we perform a similar kind of test, a t-test as before, and the mean is exactly the same because we have the same data and it just reflects the effect size averaged over participants, over all scans. But now the variability measure is different because it is based on the within subject goodness of fit. And furthermore, to calculate the standard error of the mean, we now divide by the total number of scans instead of the total number of participants, which is a much higher number. And this means that our standard error of the mean is much, much lower, leading to an extremely high t-value, and therefore a very significant effect. However, this p-value is overconfident because due to the treating of every scan as an individual data point, our standard error becomes very small, which means that we could have a tiny, tiny effect represented by a small mean, which could still be very significant just because of this. And that therefore is not recommended to use this type of analysis. Now, for fixed analysis is what we use when we compare the group effect to the within subject variability, and it's an inference about the specific sample of subjects. So you can say that if you just want to say something about the subjects that you are testing, and the statistics are often inflated. So you can't generalize to the population with these kinds of approaches. Now, in contrast to random effects analysis, you compare the group effect to the between subject variability, and it's therefore an inference about the population from which the subjects were drawn. And the idea would be that if you have a new subject from that population, you could be pretty confident that they would also show the effect if your effect is indeed significant. Now, there's also a third type of analysis, which is called mixed effects analysis. And there, some of the variables are random effects and some are fixed effects. For example, you might want to include the size of the MRI drifts that you heard about in the analysis, and you want to include that maybe as a fixed effect. And in SPM, you can do this with the function SPM_MFX. Now, everything that we talked about until now has been about one single voxel in the brain. However, usually you want to do analysis on the whole brain to find where your effect is and not select a specific voxel a priori. And the way that this is implemented in SPM is by just doing this kind of t-test in every single voxel. So we divide the brain up in different voxels and we just test our effect in every single voxel. And this is called the mass univariate approach. So this gives us an average effect size for every voxel in the brain, and it also gives you a p-value for every voxel in the brain. Yes. To summarize, for every subject and for every voxel, we take the data of the whole experiment to fit a GLM based on the experimental design. This then gives us a contrast image of the effect for every participant. And if you've designed your experiment to test for 3 or 4 different effects, you compute a contrast image separately for every subject. And this is what we call the first level analysis. So you've already heard about this in previous lectures. Now we then take these images to the second level. The single subject contrast images basically become the data for the second level analysis. So these contrast images basically become the design matrix for the second level analysis, where each row represents one subject instead of one measurement point. And we can then just do a one sample t-test to check whether the contrasts are significantly different from 0. The resulting t-values gives you a map of the effect at the group level. Now, in this specific image, the t-values are thresholded so that only significant ones are shown. So again, this is the example data set where there was a visual stimulus, so it makes sense that all of your significant effects are around the visual cortex. Now, at this point, we can insert our correction for multiple comparisons to determine the threshold for significance and ensure that our results are not just based on the fact that we are doing so many different t-tests. So because we did a random effects analysis in this specific case, we can be confident that testing another set of subjects with a similar paradigm would also give similar results in the visual cortex. So that is the summary of the random effects approach, or the summary statistic approach for testing simple effects in SPM. Now, it turns out that the summary statistic approach is not the most sensitive, which is kind of what I mentioned before, because we don't take into account within subject variance. And some subjects might be noisier than other subjects. So some subjects might have moved more in the scanner, maybe they were asleep half of the time, so that would result in a lot more variance in their responses. But in this specific way of analyzing the data, we just assume that every single subject gives as reliable data for the mean effects, for the second level effects, as every other subject. But that's, so that's obviously a big assumption. So to do this better or more sensitively, the gold standard approach that SPM would recommend is using a hierarchical model that actually takes into account both within and between subject variance, which guarantees the best results given your data. So in the summary statistic approach that I just showed you, we just took into account the between subject variance. And in the fixed effects analysis, we just took into account the within subject variance. Now, the basic idea of a hierarchical model is that the parameters of the first level are dependent on the parameters of the level above, and so forth and so on. So theta 1 at this level is determined by a combination of the parameters at this level, but also at the level above. What we are trying to estimate are the variance components at each level, and the distribution of these components are hierarchically related to each other. So you can read more about hierarchical models in this paper. Now, in our case, the first level in our hierarchical model is just the first level GLM, whereas the level above is the group level, second level GLM. So the idea is that we can influence our within level estimates by looking at our group level estimates and so forth in a hierarchical manner. So for a group level fMRI analysis, we just have 2 levels, the subject level and the group level. And here the data Y are the time series for all subjects, and the columns in the design matrix specify the design for each subject separately. So the regression coefficients correspond to the effect sizes for each subject, giving both the effect size and the variance of that effect. These effect sizes are now constrained in this model to vary around the group effect size at the second level, theta 2. And this is specified using a between subject variance parameter. And the algorithm restricted maximum likelihood allows you to estimate both within and between subject variance at the same time in an iterative fashion. And this is implemented in SPM as SPM underscore REML, rebel. And what it does is it estimates things at the second level and then goes back to the first level to adjust the estimates until it finds a good fit. And together, this ensures that both the within subject and the between subject variance is taken into account in an optimal way to estimate your effect of interest. Now, because hierarchical modeling goes back and forth between the two levels, it actually takes a long time to get an optimal estimate. So even though hierarchical model is optimal, it's computationally very intense and very slow. And in contrast, when we do the summary statistic approach, we do the first level and second level estimates separately, so there's no iteration between the two, which makes the whole process a lot faster. So in most cases, actually, we get very similar results. So for example, this is an example of an auditory experiment, and then this is the summary statistic results, and this is the hierarchical model results, and you see that's very, very similar. So given that we get pretty similar results and the summary statistics approach is much quicker, people just tend to go for this one, which is not wrong. It's just potentially a little bit suboptimal. So the two approaches are actually mathematically equivalent if these specific conditions are met. So if the within subject variances are the same, which is often not the case, but could be approximately the case, and if the number of trials within each subject are also the same. Now, even with small variations, the summary statistic approach still gives pretty similar results. Again, you can have a look at this paper if you want to know more about this. So only in cases where the within subject variance is expected to be very different do people tend to use the hierarchical model. So for example, when we look at epileptic seizures, the amount of seizures that every participant has in our experiment might be super, super different. So maybe one participant only has one seizure, one participant has like 10. So in that case, it might make a lot of sense to think about hierarchical models where you take this variation into account. So you might like to think about your own research and whether there are cases that there is a big difference in the number of trials or within subject variance between the participants, and whether hierarchical models are a better fit for your data. But in general, the summary statistics approach works pretty well. Now, all of the examples that I've given until now have just been comparing two conditions or one condition against rest in a very simple design. So to do this, we just calculated the contrast between the two conditions at the first level, and then we use this as input for a one sample t-test at the second level to infer whether the effect is significantly bigger than 0 in the group. So the example that we started with in the beginning of a visual experiment with just a visual stimulus versus rest is obviously very simple and perhaps not very interesting. So you can think of more complicated experiments where you still end up just comparing two conditions, for example, two experimental manipulations. But what if we have a slightly more sophisticated design with 3 conditions or maybe 2 groups? So the first example with 3 conditions within subjects. So every participant takes part in 3 conditions, and that means that this is a repeated measures design. For example, this could be a visual experiment with 3 different stimuli like faces, houses, and objects, or a working memory task with 1 back, 2 back, and 3 back tasks, or whatever you can think of. Now, if you want to test whether there is a difference between these conditions, we can use a one way within subject ANOVA at the second level. In general, within subject designs are much more powerful than between subject designs because we control for person-specific differences between the conditions. But if you want to instead look at differences between specific pairs of conditions, for example, faces versus houses, you can calculate a specific contrast for that comparison and then again do a one sample t-test at the group level, as we have seen before. So if not all subjects take part in all conditions, you might end up with a between subjects design. For example, if you want to test the effects of a drug, it will be hard to test that in the same subjects as you might expect the effects of the drug to linger for a while and then confine your results. Another example is if you want to compare different populations, so patients versus controls versus other types of patients. And in SPM, you can do these kinds of analysis at the second level using a one-way between subject ANOVA. Or if you only have two conditions, you can do a two-sample t-test instead of a one-sample t-test. And two samples refers to the fact that you have two different sets of participants. Now, testing for interactions can be done by specifying the specific contrasts at the first level. So for example, if you have a visual experiment where you show faces and houses for either a short time or a long time, you have two within subject variables: stimulus type, face versus house, and time, short versus long. And you might want to investigate whether the difference in neural response between faces and houses is larger if the stimuli are shown longer, and that would be an interaction between stimulus and time. So you can do this by separately calculating the difference between faces and houses for short and long presentation times, and then test those differences against each other. So all of this could be specified at the first level and then taken to the second level as a one-sample t-test again, between the contrast of the difference of the differences. Now, for between interactions, you have to do the test at the second level because a single participant does not have all the conditions. But the same logic applies where you can calculate the difference between differences to test whether there is an interaction. For between interactions, for example, a design where you want to look at condition differences between patients and controls. So for example, is there a bigger difference between seeing faces versus objects in people with and without prosopagnosia? And in that case, you specify the within subject difference, so the difference between faces and objects at the subject level. And then you do the between subject comparison at the second level as a two-sample t-test. So that is the way that you can test different types of interactions with SPM, where you use a combination of specifying some of the comparisons at the first level and some of the comparisons at the second level. Now, this afternoon, you're going to be implementing these analyses in SPM using a semantic matching fMRI task. So you'll do all different types of tests, one sample t-test, two sample t-tests, as well as this factorial design where you can test the interaction between subject effects, and you'll learn how to implement all of these in SPM. So to summarize today's lecture, group inference usually proceeds with random effects analysis, not fixed effects analysis, because we want to say something about the population and not just our specific set of participants. And group effects are compared to between rather than within subject variability in this case. Now, hierarchical models provide a gold standard for random effects group analysis because they also take into account within subject variability, but they are computationally intensive. And summary statistics are actually a robust method for random effects group analysis when the conditions are met. Now keep in mind, for all approaches, you have to do correction for multiple comparisons at the brain level if you perform this analysis for the whole brain or for multiple voxels as well. If you want to contrast two conditions within subject, you can use a one sample t-test at the second level. If you want to look at more conditions, you can use a one-way ANOVA. If you have different groups, you can use between subject ANOVAs or two sample t-test. And you will, again, as I mentioned before, try out all of these different flavors of tests this afternoon on the practical. Have fun, enjoy,